Welcome back. We're still in chapter 18 here. We're going to be picking up at this point with question number 29, which is dealing with controlled genes in eukaryotes. No operons have been found in eukaryotes. What has been found in eukaryotic genes is that they are controlled by promoters and those control elements that we'd spoke of previously. Also, these genes aren't even found on the same chromosome in many cases. Usually they're scattered about on different chromosomes. What this implies is that many genes on the different chromosomes do need to work together, and they need to work together at the same time. This occurs with genes that have a relationship in how they function, such as glucose metabolism, which is controlled by a variety of genes. Each gene necessary for glucose metabolism must be active at the same time. These activators need to make sure that all of the genes necessary for a particular function, in this case glucose metabolism, are turned on and working at the same time. This is especially important since genes that regulate glucose metabolism are not found on the same chromosome. They may be spread across several different chromosomes. So what we're looking at is different genes on different chromosomes all needed to be turned on at the exact same time so that they can do the job that they need to do hand in hand. The example I was speaking about was glucose metabolism. So looking at our question, it's asking for a plausible mechanism for the coordination of gene expression. So what would cause all of these genes to be turned on at the same time? Well, keeping in mind that we have control elements that turn on genes, we need a specific combination of control elements to turn on genes. So we're thinking that there is a specific combination of these control elements that are used to turn on the genes, all needed for the same function. Glucose metabolism, once again, was the example that I was speaking of. So we have been looking at transcription and its regulation on expression of genes, but transcription isn't the only thing that accounts for the expression of genes. Regulatory mechanisms can operate at various stages after transcription, such as mechanisms that allow cells to fine-tune gene expressions rapidly in response to environmental changes. So this would be outside factors that might influence what genes need to be expressed at a given point in a given cell. One such mechanism would be alternative RNA splicing. So what is alternative RNA splicing? We're looking at question 30 now. In alternative RNA splicing, different mRNA molecules are produced from the same primary transcript, depending on which RNA segments are treated as exons and which are treated as introns. So what is an intron and an exon is determined by what type of cell the gene happens to be found in. Cells of different organs, once again, have different functions, and therefore they need genes to be expressed differently. Therefore, cells can select different portions of a gene that are needed for that cell's specific function, and that ultimately will dictate which sections of an RNA will become an exon and which will become an intron. So it's not always the same introns in every single cell. Introns do fluctuate from one cell to the next depending on function and depending on environmental factors. Here we see an example of alternative RNA splicing using the troponin T gene, and we can see that there's different available exons numbered 1 through 5, and that will result in the primary transcript, the mRNA or pre-mRNA, that include all those. And here we can see with RNA splicing, depending on the needs of a cell, that occasionally we'll see the third exon included but the fourth excluded, and in other cases we'll see the fourth exon including and the third excluded. So here we see that both the third and fourth exons can sometimes function as an intron, and ultimately what that will create is two entirely different strands of mRNA that will produce entirely different proteins that will function entirely differently from one another. mRNA degradation, this is tied to question 31. The lifespan of mRNA is quite short in prokaryotes. In eukaryotes, the lifespan of mRNA does vary from a matter of a few hours upwards of a few weeks. One example of a long-lived mRNA in eukaryotes is the mRNA used for the development of hemoglobin. These mRNAs, they can be translated multiple times during their existence. It's not a once-and-done thing. How long an mRNA exists seems to be tied to the particular sequence of nucleotides at the end of the mRNA strand that is not translated. So keep in mind, at the end of an mRNA strand, we'll have a poly A tail, and right before that there are some other things as well that occur after the stop codon, and those are the things that will dictate how long an mRNA stays around. Does it linger, or is it broken down immediately? Moving right along, you'll notice that I've skipped over a few questions there. Those would be optional. Don't necessarily need to answer those. If you'd like to on your own, feel free. We're moving into concept 18.3 now, looking at non-coding RNAs and the roles that they have in gene expression. And these are tied to question number 35. So non-coding RNAs. It's transcribed RNA that is not translated into a protein. What is their role? We're still learning about the biological roles of RNA. What we know is that not all RNA is used in the direct regulation of gene control. What we have seen is hints is that these seemingly are not so useful sections of RNA do in fact have crucial roles in regulating gene expression, and that's done entirely behind the scenes. You could think of this as your favorite music artist who appears to be the sole person or group responsible for their music and their performances, when in fact there's many other people that are employed by those musicians that work together behind the scenes to help them have a successful career. People that I'm speaking about would be maybe songwriters who write songs. They don't actually sing them, but they do write those lyrics. Music producers and managers. You never hear about those people, but they are there. Anyhow, these non-coding RNAs appear to have influence on mRNA translation and the modification of chromatin. We will be looking at an example of these, which are known as microRNAs. So this slide and the next slide are tied to question 36, microRNAs, or miRNAs for short. Small single-stranded RNA molecules, they combine to mRNA, and these can degrade mRNA or block its translation, so they can kind of hinder it useless. Taking a look here, this is the image that you partially see in question number 36, and it's dealing with miRNA and the hairpins that they form. Keep in mind that this primary miRNA transcript, it is one single strand that's just kind of folded around because of hydrogen bonding properties, and it's called a hairpin because if you cut that off right there, that looks exactly like a hairpin. So we're looking at one hairpin right here, and we can kind of see one section of a hairpin looking a little bit closer, and this is what you have on your image. And you can see that the end over here, the bubble, there are no hydrogen bonds there, but right in here we can see that this top strand of the microRNA and the bottom strand of the microRNA, they are hydrogen bonding with one another, causing them to have this linear shape. What happens next, and this is mentioned in number 36, is the use of a dicer. The use of the dicer is essentially to snip off this unbonded end of the hairpin microRNA. So we can see it get cut off right here and right here, which would be essentially at where these little arrows are pointing at. And what's left is a single or double strand of microRNA. But if we look at it, it gets separated, and only the gold part is going into the microRNA protein complex. What will this complex do? Well, looking at it, we can see that it is bonding to the red strand here, which is mRNA. And once again, the microRNAs, they can bond to these mRNAs, and they can either block translation, so the ribosome will attach but nothing can happen. Remember, the ribosome attaches to the 5' cap, or they can break down the mRNA entirely. Moving into 18.4, we're going to be continuing to look further about differential gene expression and how this results in different cells and usage of certain genes. Keep in mind, most of this is background that we've already discussed or should be known knowledge. Everything that's alive starts off as one cell that's fertilized. So I guess you have two cells, a sperm and an egg, fertilization. And that small group of two cells combines to form many different cells. These cells are organized into tissues, and then the tissues are organized into the organs are grouped into organ systems, and then the whole organism. So all these different cells, even though they might have very different uses, they do rely on one another functioning appropriately, especially at the whole organism level, where that heart cell will rely on proper functioning of the lung cell, in essence, because we're talking about the sustenance of life here. So moving on. Embryonic development. You will know cell division as mitosis, and this is where one cell duplicates its DNA, splits in half, and we form two new daughter cells that are identical to the one original cell that we started with. We will discuss cell differentiation and morphogenesis in greater detail on the next slide, which is where you'll hear the answer to question number 38. But looking at 37, what three processes lead to the transformation of a zygote into the organism? They are these three that I have highlighted in red: cell division, cell differentiation, and morphogenesis. Expanding upon what we were sort of just talking about, we can see the fertilized eggs of a frog here. So obviously the cells in the image on the left have undergone many mitotic divisions in the course of 4 days to produce the tadpole on the right. So from this state to this state, it's 4 days. That's a lot of changing and growing and developing and specialization of cells to go from here to here in just 4 days. Something to consider is, if mitosis produces identical cells, then how do we have different cells in the tadpole? And this is the role of differentiation, which we sort of talked about with gene expression earlier, but now we're looking at it a little bit differently. Differentiation is a process that cells undergo, causing them to have unique form and function. These unique functions and shape constitute the morphogenesis of the cells, and morphogenesis translates to mean creation of form. So the terms morphogenesis and differentiation really go hand in hand. As cells differentiate, they reach their morphogenic state. Question 39 is looking at cytoplasmic determinants and inductive signals. We're going to be looking at both parts A and B of 39 here. Cytoplasmic determinants and inductive signals are used in early cell development. Consider that eggs contain RNA, they contain proteins, and they also have other items in the cytoplasm. And all of these items are necessary for the future development if fertilization occurs. These elements to the cytoplasm are not evenly distributed from one egg to the next. It's kind of random. So this does affect development. These substances found in the cytoplasm are called cytoplasmic determinants, which makes sense considering where they're found, which is the cytoplasm, and also makes sense considering what they do, which is determine development as a result of exposure or lack of exposure to the items in the cytoplasm. But not all the things that affect the development of cells are internal, such is the case with inductive signals. Inductive signals are found in the area surrounding the cell or the environment that the cell is in. Keep in mind that by the word cell, we are talking about a fertilized egg at the moment. On our next video, we'll pick up with question number 40.